In the previous video, we have discussed how the cell-cell transfers occurs via tunneling nanotube, and these are the tunneling nanotubes that we have discussed. And in this video, we will understand what are the changes occurred once the transfers occur once the transfer happened between cell to cells. So, in order to check this, in order to check the changes within the pancreatic cancer cells because of the cell-cell transfer occurred between macrophages and pancreatic cancer cells. What they have done is they have separated out the single cell colonies from this panc 0203 populations, and they have used two different populations such as SP0926 and SP1030, and similarly they did their single cell clonings for their other pancreatic cells populations that they have discussed in the supplementary figures. We will cover only the main figures that are presented in the paper. So what they have find out is that compared to the parent cells, that is pancreatic cancer cells, the SP0926 and SP1030 behave differently in a way that SP0926 are spread uniformly across the uh, area, whereas SP1030 and SP1030 forms a spheroid kind of shape, which is also in line with this panc, which is also in line with the morphology of panc 0203 cells that can be seen here. And also when they check their did the sequencing of these three cell lines, they are able to find out that all three cell lines has or all these two single colonies has the sequencing results similar to that of this panc 0203, indicating that it's the pancreatic cell lines that are spread and also acquired the macrophages characteristics because of the green fluorescence that they can because of the green fluorescence and red fluorescence these single cell colonies has, and the sorting are done based on double fluorescence signals. So first thing that they did is they decided to check cancer cell stem markers in these three different populations, and what they have identified is that in the case of SP0926, the increase in this uh, aldiflor positive cells is non-significant compared to the parent populations. However, the increase in the SP1030 is statistically significant compared to the panc 0203 cells, and these are the cancer cell stem markers. And this indicates that SP1030 has more of this tumorosphere formations. Tumorosphere is the solid sphere formation across the cells, and this is the this aldoflor is the markers for the cancer stem cells that also cause these tumorosphere formations. And this thing they have validated using this panel C, where they can see that SP1030 has more spheroid formations compared to the panc 0203, and the panc 0203 spheroid formations are in line with the SP0926 spheroid formations. And these things are also validated using the flow cytometry assay, where they can see that this DEAB negative cells, or when the cells are treated with DEAB, they can see that increase in the signals for this SP1030 is statistically significant, or it looks like it's the increase in signals for SP1030 is higher compared to the case of panc 0203 and SP0926, and these things are quantified in this bottom figure of the panel B. So after understanding that one clone has more tumorosphere formations compared to the other clones when they compared them through these parent populations of the Panc 0203. Thereafter, they decided to check their invasiveness characteristics, and when they check the invasiveness characteristics, here they find the opposite results. That is, Panc 0203 behaves similar to that of SP1030. However, SP0926 populations has more migration properties, and these things are been quantified in the bottom figure of the panel D, where they can see that SP0926 has more cell numbers in a particular area compared to the PANC0203, and the PANC0203 cells are non-significant or is similar to that of SP1030. And the one panel that I forgot to explain is the bottom figure of the panel C. So this is the quantification of the spheroids formation that are shown in the panel C. That is, in the case of SP1030, the tumorosphere formations are higher, and the diameter of tumorospheres are statistically significant in the SP1030 compared to the PANC 0203. That can be seen in this bottom figure of the panel C. The diameter of the tumorospheres are also higher, and the number of tumorosphere are also higher in SP1030. Whereas when they compare about the migration properties are higher in SP0926. So from their experiments, and they made a point that this this difference in the single cell colonies characteristics is because of the different anchorage, different kind of cell transfer occurs between different cell populations of the pancreatic cancer cells. And the two different characteristics that they have find out is this tumoral spare formations, and also and the other characteristic is this migrations or invasiveness characteristics. And similar result they have observed for the other pancreatic cell lines, which they have provided in the supplemental figures. So if you need to understand more about this experiment in detail, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.